Lab 4 Enzymes by Zuhur Hassan and Milagros Santos. Enzymes are catalysts. Catalysts are any agent that can speed up the rate of a chemical reaction without changing it permanently. They help a reaction progress 1,000,000 times faster than reactions that occur without a catalyst. A substrate is the reactant molecule that binds to an enzyme at the active site and participates in the chemical reaction. This binding of an enzyme and a substrate produces the enzyme-substrate complex. The binding between enzymes and substrates are highly specific. It is similar to the analogy of a lock and a key. The binding of an enzyme and a substrate involves conformational changes to the enzyme, and this causes it to have a tighter bond. This is called induced fit. After this conformational change takes place, the enzyme can then catalyze chemical reactions in order to form the product. In plants, catechol oxidase is a copper-containing enzyme, and what it does is it creates brown pigment when it is exposed to air and reacts with oxygen. It's the reason that fruits turn brown after they're sliced. The brown color indicates the production of benzoquinone. Benzoquinone is a substance that is toxic to food-spoiling bacteria. When the peel is damaged, oxygen can react with the catechol and protects the fruit. Preparing for the experiment. 3 test tubes were labeled 1, 2, and 3 as shown in this figure. Test tube 1 contained 1 milliliter of catechol and 1 milliliter of catechol oxidase, but no water. Test tube 2 contained 1 milliliter of water and 1 milliliter of catechol oxidase, but no catechol. Test tube 3 contained 1 milliliter of catechol, 1 milliliter of water, and 0 milliliters of catechol oxidase. The test tubes were then heated in a 37°C water bath for 10 minutes. Then we got our results. So as we can see here, the first test tube changed to a dark brownish color that indicated high enzyme activity. Test tube 2 had a little bit of color change, which indicated a lower enzyme activity. Test tube 3 had no color change, indicating no enzyme activity. Therefore, from this, we can immediately tell that test tube 1 was our positive control and test tube 2 was our negative control. For our first experiment, we tested the efficiency of enzymes at a rate of different temperatures. Our hypothesis for this experiment was that enzyme activity will be optimal at 40°C. As you can see here, as the rate of enzyme activity increases, so as the rate of the temperature increases, the rate of enzyme activity increases. Soon it will hit an optimum temperature, and then enzyme activity will start to decrease again. So our hypothesis was that at 40°C, that will be our optimum temperature. So preparing for our experiment. We prepared 6 test tubes because we wanted to test them at a rate of 6 different temperatures. As you can see here, this beaker filled with ice water was our 10°C environment. This was in a 20°C environment, and the beaker on the hot plate was our 100°C environment. We also had 3 other environments of temperatures. We had 40°C right here, 60°C over here, and 80°C over here. So prior to the experiment, our potato extract was put in ice. But if we add the potato extract and the catechol immediately, stick them in the temperature we want to test for, that would not have helped our results. So what we did was we put 1 milliliter of potato extract in each one of the tubes, and We put the tube in its designated temperature environment. For instance, test tube 1 was put in our 100 degrees Celsius environment. So we put the 1 milliliter of potato extract first, put it in the 100 degrees Celsius environment, waited until the enzyme reached that temperature, and then added the catechol oxidase to it. After that, we waited 15 minutes and we noted the color change. So we did this for each of the tubes, and on our next slide, we'll show our results. So basically, at 100 degrees Celsius here, you can tell there was no color change. At 20 degrees Celsius, there was a dark color change. At around 10 degrees Celsius here, there was little color change. At 40 degrees Celsius, there was a dark color change. At 60 degrees Celsius, there was little color change. Actually, we were supposed to have our sixth tube at 80 degrees Celsius. It's not shown in the image because there was an accident in the lab and test tube at the 80 degrees Celsius environment spilled as we were trying to remove it. However, the color had a similar color to the test tube that was incubated at 60 degrees Celsius, so it kind of looked like a faded pink color like we see here. This is our graph. As you can see here, as the temperature increased, so did enzyme activity. It hit its optimal temperature here between 20 to 40 degrees Celsius, and then after that it started to decrease. Therefore, this proves that our hypothesis was correct, that enzyme activity is more efficient at 40 degrees Celsius temperature environment. Next, we tested for the effects of pH on the enzymes. Our hypothesis for this experiment was enzyme reaction rate is fastest at a pH of 7. What we did here was we labeled 7 test tubes, 1 through 7. We added 1 milliliter of one of the pHs to each tube. For example, test tube 1 had pH of 2, so we would add 1 milliliter of the pH of pH 2 into test tube 1, and then do the same for the other remaining tubes with their designated pH. After that, we added 1 milliliter of potato extract to each tube. And then, after 5 minutes, the catechol was added into each tube. After 10 minutes, the color changes were observed. Here is our results. Test tube 1 contained a pH of 2, and it had no activity because there was no color change. Test tube 2 had a pH of 4, and it's faded pink. You can't really tell in the picture, but it was faded pink, and this indicated to us that it had little enzyme activity. Test tube 3 had a pH of 6, and you can see here that it had a darker color. Test tube 4 had a pH of 7, and test tube 5 had a pH of 8. So test tube 3, 4, and 5, you can tell that they had high enzyme activity because they had a dark color. Test tube 6 and test tube 7. Test tube 6 had a pH of 10, and test tube 7 had a pH of 12. They both have a faded brown color, indicating some enzyme activity. We can see this in our graph. According to our results, the optimal pH for enzymes would be a pH to be between 6 and 8. Our original hypothesis said that at a pH of 7, enzyme activity would be the most optimal. Therefore, our hypothesis is correct. And also, as you can see, as the pH increases, so does enzyme activity. Then it reaches its optimal pH, and then it would start to decrease again. And that's it for our presentation. Hope you enjoyed it, and thanks for watching.